Howdy, everyone. You're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we'll be talking to Dr. Dmitry Kurouski. Dr. Kurouski is an associate professor in the Department of Biochemistry and Biophysics here at A&M. He received his bachelor's and master of science at the Belarussian State University and his PhD at the State University of New York at Albany. His lab focuses on many different topics, from lipids and neurodegeneration, plant diseases, nanostructures, and forensic hair analyses. My name is Veronika, and I'll be one of your hosts today.
My name is Sam, and I'll be your co-host.
First question, how did you decide what you wanted to study when you were in school, and how has that affected the research that you do now?
Well, it's a very good question. I was always curious about life and the fundamental principles of life. So I think that's what formed my interest in high school. And I had kind of two focuses. At some point, I wanted to be a doctor, and I also was interested in more fundamental things that doctors typically don't touch, right? There's like fundamental concepts, how life is organized and why we are what we are and how our cells work. And I was influenced by my high school teacher, who was very interesting person, and she kind of biased me towards more fundamental interest, and I decided to join Department of Biology at Belarussian State University. Mm-hmm. And once I graduated, I also from the very beginning I wanted to be a professor, and to me the next step was kind of already decided. I wanted to do a PhD. And I just had to find a place where I want to stay. And I explored a few options in Germany and UK, and then this option appeared in the United States. I was invited to join grad school because the person who was finishing his PhD was also from Belarus, and his advisor asked him whether he knows someone who knows biology and who wants to work in a physical chemistry lab. And I took this challenge. It was difficult because what I did was Raman spectroscopy. I never heard about Raman spectroscopy before I joined. And also biology program and chemistry program are quite different. And the first class I took was inorganic chemistry that started from theory of group symmetry. And at some point I thought I'm in the wrong classroom because we were talking about rotational operations and objects and stuff like this. And I'm like, how does that even relate it to inorganic chemistry? But step by step, I started liking it. And also my PI gave me lots of opportunities. He was very hands-off advisor, but at the same time, very, very supportive person in research. I always knew he holds my back, and his influence, Igor Lednev, he was my PI. His influence really made me complete PhD, and I also had few research opportunities. Then I had a postdoc, and I had industrial experience, and there we go, Texas A&M.
So what kind of research did you do during your PhD, and then how did that kind of shape the way you built your lab and what y'all do?
Well, during PhD, my work was focused on neurodegeneration. We tried to understand the structural organization of amyloid fibrils. Then later, when I joined my postdoc, I had several research projects. One of them was focused on art conservation science and on fundamental physics. We try to understand the underlying molecular mechanisms of photocatalysis on mono and bimetallic nanostructures. So it was really hardcore physics. And then I worked in a pharma company where I was developing chromatographic methods and was involved in drug development. So all of these pieces I essentially brought with me to Texas A&M. And one of the projects that we work in my lab is focused on neurodegeneration. But in this project, we try to understand the effect of lipids or the role of lipids in these pathologies. And what we found is lipids do a lot. Lipids change the rate of protein aggregation, they change the structure and toxicity of aggregates and many other things, which is very exciting. But we also do a lot of this fundamental physics, specifically physics with nanoparticles. We use nanoparticles to enhance Raman scattering from molecules. And in this case, these nanoparticles are fantastically great for sensing of dyes on hair, what you guys mentioned. In this way, we advance the forensic analysis of hair. It's very important. We have funding from NIJ to do this exciting work, but also we look at the surface sites of these nanoparticles, including mono and bimetallic nanoparticles, and recently 2D dichalcogenides, and we try to understand the extent to which surface defects and surface sites change the rate of photocatalysis and many other things.
So at this point, you mentioned Raman spectroscopy a couple of times. Can you talk a bit more about what this technology is?
Sure. Raman spectroscopy is a very neat analytical technique. It is based on inelastic light scattering. So pretty much what was discovered, what was predicted in the beginning of 20th century, and what was experimentally demonstrated by C.V. Raman, an Indian researcher, is that when a light hit the molecule, there will be two types of scattering, elastic and inelastic. Elastic scattering is when photons do not change their energies, and inelastic scattering when photons would scatter back with high and low energy. And the change in the energy will directly depend on the chemical structure of the molecule. Consequently, if we catch these inelastically scattered photons and measure the change in the energy, we can predict what chemical bonds or chemical groups present in the molecule. Consequently, we can probe the chemical structure and composition of samples that we analyze. And this technique is broadly used for many different applications. Here at Texas A&M, we pioneered the use of Raman spectroscopy for agriculture. We demonstrated that we can use Raman to detect diseases in plants. And recently, we showed that this approach is translated or can be translated to animals. We predict, using Raman spectroscopy, we predict diet preferences, which is pretty cool because essentially the only thing we do, we shine laser light on mice skin. And with the collected photons, we can predict diet preferences. We can say whether mice were kept on keto diet or American diet. And we can also, for the first time, could predict the age of animals, which is a big thing in biology. So we can track the biological age using Raman spectroscopy.
That's impressive.
That's really cool.
That's amazing. Thank you.
So you mentioned too using Raman as part of forensics. Can you kind of expand on that a little bit?
Yeah, sure. So we work in forensics, we focus on two objects, hair and fabric. Hair, because hair is one of the most commonly found physical evidence at crime scenes, and it's not surprising because on average a person sheds 100 hair daily. And what we also found is many people dye their hair. And if then logic step is if we can find a technique that can detect and identify dyes present in this colored hair, we can establish a connection between suspect and a crime scene or demonstrate the absence of this connection. The question is, how do we detect the dyes that are present on the sample? It's a very difficult task because optical spectroscopy, like absorption and fluorescence, are not sensitive enough. And Raman spectroscopy by itself is also not sensitive because we deal with a very small amount of dye present on here. What we found is if we use gold nanostructures would enhance Raman scattering for molecules present on hair around 1,000,000-fold. So this 1,000,000-fold enhancement allows us to identify dyes that are present on hair at a single molecule level. And I had really talented students in my group that work on this project. Aidan Holman is one of them, Mackenzie Stachkovsky, and recently Isaac Juarez and Shannon Bober. These fantastic, talented students pushed the accuracy of identification and also were able to reveal the effect of environmental factors like UV radiation, soil, etc. on the accuracy of serous-based detection identification of dyes.
That's really interesting. I'm into true crime, and I listen to a lot of true crime podcasts. And what I do know is, previously they tried to do like microscopic hair analysis, right? That they kind of ruled that as a junk science, basically, right?
Correct.
And so finding hair at a crime scene is really only beneficial if the root, is there, right?
Right.
But this could be really cool for samples that don't have the root attached. They can't do a DNA analysis. So it just opens up a whole new avenue for forensic technology. I think that's really cool.
Thank you. Exactly. It's exactly true. And we got funded by National Institute on Justice. It's a very competitive funding opportunity, and I'm very proud that we received this funding. And it's a great institution. They invite us to their conferences, which is American Association of Forensic Sciences and PeatCon, to present our most advanced findings on this topic. And at this moment, we're also interested in fabric, because obviously all our clothes are colored, and we think that this technology can be used to detect and identify dyes present in our fabric, and pretty much also help the forensic experts to make their work easier and more reliable when they make conclusions.
It's really cool. Yeah.
So have you always wanted to be a professor?
No. No. At some point, it was my postdoc. I had really challenging postdoc because first we were funded by Air Force, and it was a very difficult project. We had to explore nanoelectrochemistry, and we had to work very hard because we had to report our progress every 6 months. Mm-hmm. And at that time, the academic market was very saturated, oversaturated. And I had colleagues in my group that applied 2-3 years before they got the academic position. And at that time, I started thinking about alternatives. And right at that time, a very prestigious and very well-established company, pharmaceutical company, Boehringer Ingelheim, came to me and they asked if I want 6-digit paycheck. And I said yes. I said yes. I was a poor postdoc in Chicago, and having this financial opportunity, I was like, wow. So at this point, I switched my interest and I joined a pharmaceutical company. It was great. I should tell you, I really enjoyed working there. But in about a year, I got bored, because working in a big company, you do the same thing every day. And at some point, you start feeling that if you don't come to work, nothing will change. So at that point, I realized, no, I want the challenge. I want something interesting, something that would make me wake up every morning and want to come to work. And the only job that fits this criteria is academic job, in my opinion. It has its own challenging moments. I don't want to hide it. It's not a perfect job from perspective of work-life balance, but it gives this freedom to investigate topics that you are interested in, do the work that you are interested in, compare it to industrial jobs.
So what made you, when you were switching to professor, what made you choose A&M as well, and then eventually join the gene program?
Very good question. So A&M had very interesting and yet very weird opportunity. So they had a hire for a biophotonics institute. So Biophotonic Institute, it's called IQSC, Institute for Quantum Science and Engineering. It's a virtual program similar to genetics, and the program meant to find people with a strong background in optics and sensing and spectroscopy and everything in between, and make them work with biologists. So, and it was exactly what I was, because I had a biological background, and at the same time, I was a spectroscopist. So looking at this opportunity, I said, well, it's perfect. It's exactly what I want to do. And it actually was a really great idea to launch this program, because once I appeared in the biochemistry department, I realized 2/3 of people in that department want to work with me, because I have these great techniques, great tools, and they have lots of very interesting questions. And in fact, I worked not only with people, collaborated not only with people in my department, but also probably with half of departments in Aggie College. I worked with people in plant pathology, microbiology, soil and crop sciences, horticulture, poultry science, you name it. And because of these diverse collaborations, I was interested in many programs, including toxicology and genetics. But the reason I got in the genetics program was completely different. I had a very talented student, Samantha Higgins, and she joined genetics as a PhD student. And she wanted to be in my lab because she did undergrad in my lab. She was one of the most talented and super organized students that I ever mentored. And because she wanted to join my lab as a graduate student, I joined genetics, so I could be a formal mentor of Samantha Higgins. And I was very surprised that after one of the rotations, she decided not to join my lab. And it was not very surprising to me at the same time, because I realized that she wants to do something else. And she was very interested in the mice project. and she studied the effect of alcohol on birth defects, specifically male-triggered birth defects. And it's very cool. But that's how I joined genetics, not entirely because I'm a geneticist, but because I wanted to support my student.
Yeah.
I didn't know about that.
Yes.
Because Sam is a year above us, I think, right? In that cohort?
Correct.
Yes.
I actually knew her in undergrad as well. We took some classes together. So I've known her for a while. Yeah.
She's an Aggie. She's
Yeah. So part of being professor is teaching. So how do you feel about that and incorporating that into, just like your day-to-day life?
I love teaching. I truly like teaching. I teach 2 classes. I teach for 10, which is very popular. It's a very big class. I teach it in the fall. It's 150 student class. It covers the fundamental topics in biochemistry. We start from amino acids, then we go to proteins and sugars and enzymes, etc. And it's very interesting class because students have to go through many topics, and at the same time, everything that they go through is very applied. So to come to that point, they took already Gen Chem, they took Orgo, and now they're very excited and very mature to study this concept. And also, I feel that they really enjoy because they can apply them. Unlike in organic chemistry, all these mechanisms, it's really hard to apply to real life. Biochemistry is very applied. And I also teach a module for graduate student in biochemistry department in a spring semester that is focused on optical techniques. So in this module, we discuss absorption, fluorescent, infrared, Raman in a context of how it can be applied in biology. So I train students how to use these techniques, what information they provide, and how to avoid getting artifacts in their data.
Yeah. It's interesting you said that. I did actually take you for 410, and that is exactly my experience.
Thank you.
I will be honest. I hated organic chemistry.
I loved organic chemistry. Okay?
It felt imaginary to me. Like I can't see it. It didn't necessarily click for me. But biochemistry, I've always been more interested in the biological side of things, right? And so biochemistry felt significantly more applicable and interesting. And I did appreciate taking your class. So,
Thank you.
Yeah.
Thank you. It was great having you as a student.
If you remember.
I do, of course. I do. By the time, so I remember many students, and I stay connected with many students that took my class like 5-6 years ago. Some of them work in my lab because I typically invite several students to join my lab, and others just send me cards or emails from time to time, and it's very nice. It's really this moment of appreciation that it means a lot to me. So yeah.
I like that you're able to connect with students even at such a big university, because I did my undergrad at a small university and it was pretty easy to connect with my professors. So I also stay in touch with them. I have them like on LinkedIn, and I already invited one of my professors here to do a seminar. So I love being in touch with them. But during your studies, when you were a grad student or even an undergrad, did you also go to like such a large university like Texas A&M?
Yes.
Yeah.
Yes, I studied at Belarussian State University. It's the biggest university in the country. And I had, so we have a little bit different system there. It's not like every class you take with different people. So you start with the same cohort of people. So our class was 100 people. And then you split in subgroups, but to a large extent, all major classes you take with 100 kids. So yes, it was very similar to this. I'm from a very small city. I was born in a town that has 20,000 people. But yes, once I moved to the capital of Belarus, to Minsk, I was in a big school. Yeah, but there was one very interesting thing that you mentioned. Yes, indeed, it's a problem that we have very big classes, and to a large extent, students do not feel this connection between them and professors. They think that professors don't really care, professors don't have time. And I can tell you one very interesting moment. I saw a student, so I'm on a second floor in Department of Biochemistry and Biophysics, and that's a place where we have our biochemistry and genetic labs. And once coming from lunch, I saw a student on the corridor, and I said like, "Hey, John, how are you?" And he turned to me and said, "How do you know who I am?" I said, because you're in my 410. And he said, and you know my name? I said, yes. And he said, look, listen, I'm a senior, so I've been here 4 years, and it's the first time the professor knows my name. So I was really like, wow, are we that bad actually? But so in my class, I make an effort to know students personally. Some of them come to my office and ask questions for office hours. But even beyond this, I ask students in the class to participate. I often call them by names and ask them to come to the board and draw something. So yeah, I care about that.
That's really nice to hear.
It's
especially
really...
because I did talk to some undergrad students here who did say that the classes are really big, and I never experienced that, so I can't even imagine. But they do say that they obviously feel a big disconnect with their professors, which is something I didn't experience, but you're obviously making a difference.
I'm trying.
So you mentioned that you're part of multiple different departments, right? Including like biochemistry, chemistry, genetics. So can you maybe just briefly discuss some of the different projects that you do with the different departments?
I have several different projects in my group that resonate with some programs or departments. I mentioned forensics projects, so I'm also in a forensics project. I'm also in a forensics program at Texas A&M. We have a very small forensics program that is run by entomologists. And it's not surprising because a lot of work in forensics is done by forensic entomologists. So they come to body farms and they use maggots to predict PMI, postmortem intervals. And by the way, we have 2 body farms in the state of Texas, one in San Marcos and one in close to Huntsville.
Can you elaborate what a body farm is?
Yeah, sure. So the body farm, it's a research facility. It's BL2 research facility, so you cannot come there in shorts and flip-flops. It's in San Marcos. It's the small area in the ranch that is gated, so it has a fence around, so you cannot really get in there very, very easily. You have to have a permission. And we work with Texas State that has a department of forensic anthropology and Professor Daniel Vascott. So we work directly with him to get the access to the body farm. Once in there, you see bodies that are placed in different environments, in shaded environments, in open sun environments, and forensic anthropologists do a lot of crazy research. They burn bodies, they do a variety of different, like hang bodies, because one of the bodies, when I visited, was hanged, and we asked what's going on, and they said the army was very interested in this research because of the suicides that happen, I think, to some extent in the army, like soldiers hang themselves. So army was interested to track changes that take place in that type of a body at different time points after the death. To a large extent, all this work is done to predict the time of death, to predict how long the body was on the ground, under the ground, in different environments, because it's very important for the forensic experts. And my group, we do experiment there right now. We just finished one experiment in which we investigated the effect of decomposition fluids. It's the fluids that leave our body once we decompose, on the accuracy of detection of artificial colorants, dyes on hair. So I have a student, I mentioned his name, Aidan Holman. He colored hair in the lab, and then he brought all this different hair samples to the body farm, and the student at Texas Tech, she collects hair at different time points and then sends them back to us.
So impressive.
Yeah.
I didn't even know that facilities like this exist. I mean, as you mentioned, research in this area is very important to understand even just the time and the causes on a different level, but I never thought about it.
Mm-hmm.
Sorry.
Please go ahead.
I know you're not from Texas. If you did drive past it, you can tell.
Oh, so you know.
Because you can smell it. Yeah. Because it's a bunch of decomposing bodies.
Yeah. This smells is something that you need to be aware once you visit, especially during the summer. My first visit there was in July, and I think they had like 30 bodies, so you can imagine the atmosphere when you're in a hot, sunny day with 100 degrees outside, which is already enough for me, for the northern Belarusian guy. Yeah. And when you have this type of smells, and
that's wild.
It's intense.
I'm so intrigued by this.
Right, but we also do fundamental work. We work in the area of fundamental biochemistry and genetics, and here we do lots of very cool stuff. I mentioned this work on protein aggregation. So we use live models. We use C. elegans to understand the effect of lipids. So we grow C. elegans on different media with different lipids, and we try to find the extent to which different lipids change the lifespan of C. elegans, and that's very interesting. It gives us this really fundamental knowledge about nutrition and about the extent to which our nutritional preferences change or may influence chronic diseases. In my opinion, it's very important. And I also have a very talented student, Charles Mitchell, who discovered that 2D metaldehyalkogenate nanoflowers can be used to extend the lifespan of worms and reverse detrimental effect of Parkinson diseases. We have C. elegans that overexpress alpha-synuclein, and they develop the Parkinson symptoms, so they don't have strong thrashing, and they have much shorter lifespan. So with his great work shows that these nanomaterials, they expand the life of C. elegans and completely alleviate symptoms that are caused by Parkinson, specifically induced by alpha-synuclein aggregates. And it's very exciting because it's the first evidence of a neuroprotective effect of a drug, and because everything we have to this point only alleviates the symptoms. Nothing is neuroprotective. So this is very exciting. And we have several other projects in the lab that we're also very interested in immune cells. We try to develop immunotherapy to treat neurodegenerative diseases. We program macrophages and NK cells so they can get into these inflammation places and degrade protein aggregates that cause, as we expect, neurodegeneration.
Very cool. Yeah.
So is Parkinson's disease the only neurodegenerative disease that you focus on, or?
No, we focus on several. We work with on Alzheimer's disease. We also Parkinson is one of them. We work on diabetes type 2. It's not neurodegenerative disease, but it's amyloid disease. And we focus on tauopathies. It's a group of diseases that are linked to aggregation of tau proteins in the brain.
You said type 2 diabetes
Correct.
is related to amyloid?
Right.
I did not know that. Can you explain that a little bit?
Yeah. So type 2 diabetes is a disease that is linked to aggregation of amylin in pancreas. And these aggregates are very toxic, so they destroy beta cells that produce insulin, and that's what is causing diabetes type 2. And in my lab, we found that lipids drastically change aggregation properties of amylin. Some lipids make it less toxic, some lipids make it more toxic. And to a large extent, the effect of lipids was unclear and unknown in terms of aggregation of this protein for decades. And it's something that we discovered very recently. I have a very talented student, Jaden Seaton. He leads this project. His paper just got accepted to GBC, and it's something that we're all very proud of.
Congrats. That's very cool. Yeah, I did not know that.
Thank you. Yeah.
So I'm going to switch up the questions a bit from research, but focusing more on mentoring.
Sure.
So how do you mentor your grad students? I'm even curious, how big is your lab? So how do you manage having everybody in the lab?
Good question. So my lab is about 7 people, and I think I have a good balance between young researchers and accomplished researchers. I have 2 really experienced senior research scientists, and they have one of them, Yana Purvish. She's an expert in genetics, and second researcher, Mikhail Matvienko. He is an expert in cell biology, and they really help PhD students, especially young PhD students, to learn the methods and to get this experience. But I also think PhD students is the focal point of the lab. They're really the driver of research. They have this energy, and they're a vital component of the lab. But I also have a lot of undergrads. So my lab is about 15, 17 undergrads. We have a lot. right? An army of undergrads. These guys are really fantastic. Many undergrads in my lab publish papers and publish papers first author, and many of them work very hard, exceptionally hard. So I'm very proud of them. And of course, my mentoring style is a little bit different for these different groups of people. If we talk about PhD students, I always tell, so PhD students join the lab in January, the year one after rotations, and I always tell them that they need to learn methods that are necessary for their independent performance for their PhD by the end of the semester. And of course, by learning, as a lab, we train them. As a lab, they work with senior students and research associates to learn and advance their skills and their techniques. And I may give them a few months during the summer if I see that maybe teaching was taking a lot of time and classes were taking a lot of time. But that's my expectation. And I'm very honest and upfront and telling that, look, if you could not learn and be independent in 3-4 months, then you probably should look for another lab. And I have students leaving my lab, so it's perfectly fine. After this, I also, during this time and after this first semester, I meet with my students biweekly or monthly. And in these one-on-one meetings, we discuss the progress and we try to understand what worked, what didn't work. And I think I'm always constructive. I'm always helpful. But at the same time, I'm honest, because what I think students should know is how well do they do. Because if they don't get this feedback, frustration builds up, and this frustration often kills the motivation. So I think, if there is no progress, student needs to know that there is no progress. And obviously, I have to show the avenues that the student need to explore to get stuff done. And in my group, guys present approximately once in 2 months in front of everyone, so that also helps them shaping up. And all this together, I think it's my mentoring strategy. As a person, I think I always prefer carrots over sticks. So I always try to encourage and motivate students rather than punish. I'm never like the guy who's like, no, you have to stay in the lab and get this done. I'm always giving opportunities and encourage students to explore them, to try new things, to explore these uncharted territories, because to a large extent, many talented students came to Texas A&M to do this work because they're very excited, they're energized, and they're fearless. They want to study really cool things and spend the time of their life to answer these questions. And I think as mentors, we have to help them, providing this training and also providing this mental support. Essentially, I always try to show my students that I will hold their back, that I will not be the person that would yell on them because something didn't work. Because research is a challenging thing. We do things that were never done before. And I think that altogether, it's how would I summarize my mentoring strategy? Mm-hmm. But you better talk to my students. I think that
maybe
would
they have
be
something different to say.
Maybe they have something different to say. That's right.
So I guess along the same lines, you have a lot of different research projects going on. Right. So how do you kind of manage and compartmentalize all those different projects, but also stay up to date on the body of literature for each of those different topics? Great
question. It's very difficult. I indeed have a lot of projects, and I have a student right now who works on plants, and Isaac Juarez, he's about to graduate. He studies the effect of different toxins on rice, aiming to develop Raman spectroscopy to detect this type of stresses. And what I can say is, to a large extent, the conversation between me and a student is the drive for new ideas. It's very interesting. I sit down with a student and we start talking about science. And what is very important in this way is we're not looking at each other. We're looking in one direction. We're not trying to please each other. We try to conceptualize our thoughts and see what's the next step. And often next step, the concept of the next step makes both of us go and read about it. We're like, oh, why won't we, do you think we can do that? We're like, that's a cool idea. Let's read about it. So I read and the student reads. And then we get together again and like, well, maybe we need to change a little bit the focus or consider a few other things that were or were not reported in the literature. But you're completely correct. It's very difficult. Sometimes I feel I go crazy because indeed I have like 5-6 projects in the lab, and sometimes I go to bed at like 2 a.m. or even later because I have to. Some of them are more easy, but others we recently finished a very successful project with the Department of Homeland Security. We were developing a sensor that can be used to detect and identify pathogens, including viruses, and we had to report our progress every month. So, of course, that was very difficult. And students that work on this project were great, but in the same time, we had to be very organized, like Swiss clock, to deliver every single month and please these people. It was a big success, and we expect to have a second phase that we still discuss with Department of Homeland Security for this project.
You said report once a month?
Once a month.
Wow.
Yes.
That's a lot.
Right. You can imagine the tempo.
Yeah.
We in general we live in this Allegro, but for this project it was
So
Allegro vivace.
there's so when you become a PhD student, you get asked if you want to potentially end up in academia or industry. So students that complete research and complete their PhDs in your lab, where do they usually end up? And if they already have like a selected path that they want to be on, do you mentor them maybe differently? Do you potentially like push them in different ways? Like, can you discuss that?
Yeah, good question. So I graduated 2 students. Both of them are in industry. Both of them work as engineers at Brooklyn Nano. So they live pretty good life. One of them is in California, another one is in Boston. And the truth is that 7 or 8 out of 10 students in our university pursue industrial careers. So that's the truth. So we have very small number of students that want to be professors and become professors. And I don't really have different mentoring strategies, but I always tell that they need to look at industry. And I, as a person who works in a pharma company, I know very well what is industry, and I can advise them what is good and what is bad in industrial jobs. I tell them, yes, it's a nice work balance. It's 9-to-5 job, but there are some disadvantages. You may get bored, or you may feel you do the same thing over and over again. But it's ultimately their decision what they want to do. I'm a little bit harder on students that want to pursue academic careers. I have a student, I tried to convince him to become a professor, Charles Mitchell. I think he's old, but every single thing that he needs to become a professor. And I'm a bit harder on him because I think that academic jobs are much harder to get. They're much more competitive than industrial jobs. But of course, I respect the choice of a student. I respect we're all very different. Some people want academic jobs. I have a student who graduates, he wants to be a pilot, Isaac Juarez. He graduates in 3 years. It's the shortest time to degree I've ever seen. And he does that to become a pilot. And he ultimately wants to work for NASA and work up in a space station to do biological experiments. So I have my group is very diverse.
Sounds like
and I have a student, Aidan Holman. He wants to work in a government type of organization once he graduates. So again, I respect their choices, their vision of the world, and I try to help them come to this, be successful in these careers that they want to pursue.
So how do you also kind of mentor and prepare your students as they are writing papers or getting ready to do presentations, presenting their research to diverse groups of people, diverse backgrounds as far as like, what kind of research that they've been involved in?
Yeah, so I strongly encourage them to present. I think it's very important and necessary part of the academic training. Many professors overlook this, which is very unfortunate because many people think about PhD as a lab work or bench work. It's just one of the components. Another very important component is going and presenting. And I encourage all my students, both graduate and undergraduate, go to Student Research Week that we have here at Texas A&M and present and compete, because there are prizes. And also all my students presented there. All my students present at the symposium that we have to recruit students. We have this joint venue that is on Friday between genetics, toxicology, neuroscience, and biochemistry. So all my students this year presented there, poster or oral talks, and I think it's very important because it allows the student to get the feedback, and I think it's very important. And also, I send all my students to conferences. It's expensive for the group. For example, Charles Mitchell last year went to 2 conferences. He presented at FASIB meeting, and I invited him to present at the Amyloid Symposium that was held in Tallahassee, Florida. These meetings are very important to get the feedback from the professionals, from people in this research community. And it's completely different feedback that students get at Student Research Week that is much more general audience. And I think all these pieces help students to shape up their research, because my students publish a lot, and I think that very quickly they think that they're the best, but they're the best in their cohort of people. And I always tell them that don't look at people from your cohort, because you will compete against people from MIT, from Harvard, from Northwestern, from Stanford, from Peking University. So you need to see these people. They're very good. And these conferences give them opportunity to see these people, to compare themselves, not against people that they will not compete, right? But against their future competitors for academic job, for industrial jobs. And I also can tell you that this conference has really transformed students' vision. They come back very energized, very enthusiastic, with a lot of ideas, and they're very excited to test these ideas. And I think that's very important. It's a very important part of the training, PhD training.
Mm-hmm. Yeah. So part of conferences too is also networking, which right. I think is not something that I've necessarily discussed extensively with like my PI or anything like that. But how do you prepare your students for that? And if so, how do you kind of go about that?
I not only prepare, I actually participate in networking. So for example, I go every year to a meeting that is called SciEx. It's Scientific Exchange. It's the annual meeting of Applied Spectroscopy, Society of Applied Spectroscopy. And there are many spectroscopists come to this venue. And for students that I take with me, last year I took 2 students with me, I virtually grab their hands and walk around the hallways, introducing them to my colleagues from University of Chicago, from ETH Zurich, from Stanford, etc. I know many people in these venues, and I help my students to network, because typically it's harder for students to come and shake someone's hand. Students are very reserved, and I think the PI should play a role in this type of introductions and help students to cross this barrier. So that's what I do to my students. I go there with them, and I can help them networking. And it helps, because these connections play very important role, if not the key role, in industrial jobs. Because in industrial jobs, connections is everything.
Right.
So we have this question that we ask all the guests that we have on the podcast, and that is, what do you do in your free time to stay sane?
Good question. I like traveling. First, I don't have a ton of free time. When you do? When I do, yes. Well, I try to keep myself in shape, so I try to work out a little bit and swim. Sometimes I can do it a few times a week. Some weeks I cannot do it at all. I like cycling, I like biking, and I like traveling. So in my free time, I try to travel, because the world is really beautiful and very interesting. There are so many interesting places. This January, for example, I went to Ecuador and I went to the jungles. So it was super cool. It was absolutely stunning experience. So I saw monkeys and caimans and you name it, every single thing that you can see in a rainforest. It was absolutely gorgeous. And I also hiked in mainland Ecuador, and they have very nice city. It's called Baños in the center of Ecuador. I rented the car and I was by myself. I went to a few volcanoes, Cotopaxi and Chimborazo. and many other interesting places. So I try to have several trips like this every year and explore the world.
That's very amazing.
Yeah, that's really cool.
I think sometimes PIs forget to take some time off maybe and go travel and see the world. So it's good that you do that.
Thank you.
What's been your favorite place that you visited so far?
It's hard to say. In the United States, Death Valley, it was the place that surprised me the most by first and foremost absence of a green color. You do not see green. So I remember first time I saw green field after a week in Death Valley, I was surprised, like, wow, the green is so beautiful. And also the scale of that park. You drive like 50 miles across the valley, you get up in the mountains, you cross the path, and you see another 50 miles. And you do it 3 or 4 times, and you feel insane because you feel that you have seen it already. So that was one of the most interesting places in the United States. Europe is also a very interesting place in terms of like cities, I would say. There are many beautiful parks in the US. There are many beautiful cities in Europe. Prague is one of them. Of course, Paris and Amsterdam, London, many other great cities that are very interesting. And it's hard to find like one particularly amazing place. I feel comfortable in many places in Europe, and I feel really excited in many places in the United States. I recently visited the Big Bend, which is like 15 hours driving.
I need to go.
Yeah.
Yeah,
but it's so far away.
That's exactly what I was saying myself every year. So it's my 8th year in Texas, and every single year I heard that it's a great park, and it's exactly what I said to myself. It's like freaking 15 hours of driving, and I'm like,
and you're still in Texas.
And you're still in Texas. That's
Yes.
precisely correct. It's absolutely worth going. It's very. I stayed there for a night. It was great. For 2 nights actually.
In Texas, Paladuro Canyon is also really cool.
That's right.
Yeah.
That's right. It's on my to-do list.
Yeah, and they have the musical set in the canyon that you can go to, which is actually really cool. I've been twice.
Right.
So, yeah.
I need to travel more around Texas.
It's hard as a graduate student to take any time off and
not
do
feel guilty
anything.
about it.
Yeah.
Well, what I always tell to my students that they always have to balance. I do not expect them to work more than 8 hours, and it also shouldn't be flat. I think if the student comes and works precisely 8 hours a day, it's not good. So there are obviously like up and downs. You know, there are moments where you have to stay until midnight and past midnight in the lab to finish the experiment, but there are obviously time when you can just relax. And in these moments, including holidays, travel. I traveled a lot while I was in grad school. I traveled much more than I travel now. Maybe because I was very motivated and excited to see many places in the United States. So yeah, absolutely do it. You're young, you're enthusiastic, you're energized, and you don't have too many responsibilities yet. Beyond the lab, there is no much on your plate. So,
Yeah.
so enjoy your life.
Sometimes the lab feels like a big responsibility.
It is.
Yeah.
But learning how to balance, it's very important. It's very important in grad school.
Yeah.
Is there any advice that you would give to future grad students?
Well, good question. I would say listen to your heart when you choose your future. Do not go after shiny things that are modern, that are very interesting at this moment, especially if you don't like them, because you will not enjoy it. You will not be happy. Do what you're excited about, and you will see that even if it's not super cool topic, you can make a difference. You can be the one that will transform the world because you care about this, because you're excited about this. So that's an advice I would give to all students. Because another advice I would give is use this time that you have wisely, because obviously students spend a lot of time for things that don't really advance them. And I personally think that having this focus is very important, because at the end of your PhD, whatever, it's 4 or 5 years, you will be over, and this time in your life in which you can do lots of exciting things will be over, and you would be like, could I do more? Probably yes. So, but you will not be able to return this time, to return this opportunity when you are free, when you're excited, when you have lots of opportunities to do cool stuff, to learn cool stuff. At that time, this will be over. And I know many students regret. Many students think, damn it, why was I fooling around? I could have done so many cool things. I could have learned many techniques. And they regret at that time point. So knowing this in advance is very important.
Thank you so much for coming and joining us on the podcast. This has been very interesting.
It has been very, very interesting. I learned so much.
Thank you for having me. It's a pleasure to talk to very enthusiastic, very smart students. I think you guys do very important job, not only because you make science visible, but because you make science interesting. And I hope that your interviews, your videos, and your work will attract more students to graduate programs, because we need talented students. Unfortunately, many students that are very talented, they pursue other careers like medical and dental careers. And obviously, we have to work harder to make science more interesting in our society, to attract young minds and show them how exciting and interesting and challenging this life path is. So thank you for having me.
Thank you. Thank you so much. Today's episode was hosted by Samantha Foster and Veronika Mojic and produced by Anthony Matarazzo. The podcast is brought to you by Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, and X at Genetics Podcast. Thank you for listening. We hope you'll tune in next time.